The techno, ¿verdad?
Good evening, everybody. Thank you all for being here. Today we have this international collaboration, which for me, for us, I think that is very special. RSG-Spain and RSG-Iran have joined forces to try to create an international seminar and also try to make it intergenerational, and in this case even multidisciplinary, because we have today two amazing bioinformaticians. The first one that was invited by RSG Spain is Alicia Gómez González, and she will start today. I will give you a brief description about her in a few minutes, and then it will continue Dr. Ali Sharifi Zarchi, who will be presented by Hossein Al-Attadi. And well, for you to have one idea on how this activity is gonna it's gonna be developed. It's gonna be 20 minutes presentation of Alicia talking about how she got into industry, what she does in industry, what are the current demands in this field, and then the experience of Dr. Ali Sharif will be also shared by everybody in 20 minutes for each one. And then we will provide an extra 20 minutes for everybody to launch questions regarding on how was bioinformatics, how is bioinformatics today, what do we need to learn, what do we need to focus more. So to go directly into matter, Alicia Gómez González did a master's in bioinformatics. She has been working for 3 years in industry, and she's mainly specialized in genomics, bioinformatics in health. Alicia works in the Spanish next generation sequencing industry. This business is called Dreamgenics. Where is located? It's in
the north of Spain, in Oviedo.
In Oviedo, in Asturias, very nice. Well, she mainly works analyzing genomics data for clinical and research purposes, which means that at the end, in bioinformatics, many researchers and professionals, at the end, we do many things the same. We are going to discover that in a moment. So she recently is involved in cancer-related projects and in a clinical trial with a pharmaceutical company. If you want, Hossein, you can introduce Dr. Ali Sharif, and then Alicia, you can start your presentation.
Of course. Hello, everybody, and good evening. It's an honor for us to have this collaboration with the RSG Spain, and it's the beginning of our participation and our collaboration in different areas. And hopefully we have two very great esteemed presenters, Dr. Ali Sharifi and dear Alicia. Dr. Ali Sharifi, he's assistant professor of Sharifi University of Technology, very well-known universities in Iran, and his expertise in computational biology and bioinformatics. And we have the privilege in Iran that we have many diverse courses and different courses in bioinformatics from Professor Sharifi, which especially at the moment, we have a very broad and the course of machine learning in Iran that many students all over the world are participating in this course in Persian language. And it's an honor for us to have Professor Sharifi's advisor in RSG-Iran and also in Royal Bioinformatics group. Also, I can mention that Professor Sharifi, he's the chairman of International Olympiad in Artificial Intelligence, and also from the previous honors, the gold medalist of International Olympiad of Informatics. And today we have the honor to have his speech on academic side, academic aspects of bioinformatics and importance, and how we can maybe in the discussion we know that how we can relate and connect the academia and the industry together. Thank you.
Thanks so much, Hossein. And Alicia, you can start telling about you.
Okay, so I will share with you my screen. Okay, so good evening, everyone. As Adrian has already presented me, thank you very much. My name is Alicia, and I'm a young bioinformatician with 3 years experience. Even though my journey was still not long, I want to show you like how my career was focused, because maybe it would be first inspiration for some of you. First, I would like to highlight that my bachelor is not the typical one of maybe biology or technology one that is usually the start of a bioinformatician. Instead of that, I did a bachelor in food science. There I discovered that I really liked genetics and biology, so I did a master in genetics later. I wasn't sure about doing a PhD, so in the meanwhile, while thinking about that, I tried at the industry in a project management position. There I show a little bit how industry was working, which things are important for them, how communication between different businesses is going on. But in the end, I still wanted to work with data, work with biology, so I moved again to a molecular biology lab. And there I asked myself, why not to try data analysis? That is something really growing and there's real need for it. And well, I can say I really like it because I tried the master in bioinformatics, I loved it, and then I started my current position as bioinformatician. So now that you know a little bit about me, it's time to get to know Dreamgenics, the company where I work. As Adrian has already said, we provide bioinformatics solutions both for the clinic and both for researchers. We provide services, so we just want to contribute to precision medicine. Also, for you to know, it's not an old company. It was born in 2011 as a spin-off of the University of Oviedo. So again, maybe like the importance of research, because things like industry can come from that. We have our own proprietary software to carry out that NGS data analysis. And later on, we also open our own laboratory and genetic counseling department. Early this year, we developed also a sequencing platform so we can offer sequencing to our clients. Just for you to have some facts about the company, we have a 12 years experience in NGS analysis. As I said, we own the proprietary analysis software. This comes with in vitro diagnosis certification, so it can be used for diagnose. And we deliver our results in a platform we call Genome One Reports. I will talk a little bit about that later. And I would like to highlight the importance of quality certifications. At the moment, we count with 3 of them. That is a quality management system, quality management in medical devices, and information security management systems. I think in industry, having different certifications to really prove like you go through different processes in a good way is important. About what we offered as an NGS company, we have to be able to help our clients to understand which type of analysis they should do with their data according to their needs. And also we can help them in the process in case they don't have a lab or a sequencing platform. But in the end, what our main goal is to do the bioinformatic analysis for them. Having known all of that, which type of analysis we carry out in our company? Well, we can group it in 3 different big areas. That would be genomics analysis, transcriptomics, and multiomics. On the first one, we mainly carry out genome, exome, gene panels, trio, multisample, and somatic versus germinal analysis. On transcriptomics, we have experience on RNA-seq, microRNA-seq, and single cell, but now we are also developing spatial transcriptomics. And last but not least, in multiomics, we carry out integration of transcriptomics, proteomics, and metabolomics data. And myself, I'm inside the genomics area, so on the further slides, I will just speak on that point. I will first introduce you to how a typical NGS pipeline for genomics is. It would be a simplistic one, but just for you to know like the main steps, in case you don't know, you're not familiar with it. To start with the secondary analysis, you have to receive the raw data from the sequencer, and you first need to do a quality control of it. Later on, you need to carry out the trimming to remove both adapters and also bases of low quality. Later on, we mainly do read alignment using the well-known aligner BW-MEM. According to the necessity of the experiment, if there are a lot of read duplicates, they need to be removed. For that purpose, we use some Bamba tool. And last, on the variant calling process, we carry out two separate processes, both of them using in-house algorithms. And first one, trying to detect small variations like SMVs and indels, we use a caller we call digicaller. And on the other way, we do copy number variations using capture CMV algorithm. Last, on the tertiary analysis, focusing that this type of analysis are mainly for diagnose or research projects where they need to detect concrete variants. We carry out also annotation using an in-house algorithm. There, we annotate information from several databases, both populational, clinical databases, or in silico predictors. And we also add a context to the variant. That means we add several statistics and information so the analyst can't really see if it is a true variant or maybe just an artifact from the sequencing process. Now that we know like the structure of the analysis, I want to show you how these results look like. As I said before, we have a platform where we deliver the result in an interactive way for the customer. It is accessible from the web, and you get to see like interactable figures and tables that you can export them for your papers or your projects, and you have plenty of filtering possibilities. Taking as an example the results of an exome analysis, I will show you how it looks like. On the first side, I would like to highlight that it's important when you are reviewing or analyzing an NGS project like this to see the statistics from the sequencing process. You need to see if the coverage of the analysis was good, if your probe is having efficiently, I mean, like if you don't have a lot of gaps over probe and you are getting the coverage you expect in all the regions of the exome in this case, if you have many duplicated reads, etc. For that, we also have some tables or indexes that we call calability indexes, where we can get to see like those regions, those concrete regions, and how the coverage, like the minimum coverage that was reached on that region, the maximum coverage, etc. We can have it for each gene or each region of the probe. Then about the variant index, it is an interactive index with all the annotations both on the columns, and also we have what we call facets that you can see it on the left. There you can get to see all the variants that were called in analysis and see different information like zygosity, protein effect, the chromosome and the change, different statistics like the number of reads with the reference allele, number of reads with alternate allele, etc. And you can get to filter them very easily to finally get just your variants of interest according to the experiment. About that, you can also add a review to the variant. That means once you have your variants of interest, you can select a concrete variant, open this window, and you can add a note, or you can add, for example, like concrete ACMG criteria that you have observed. Maybe this could be more interested from a clinical point of view, more than research, but it's facility you have. And then, for example, with this, that variant keeps saved on a kind of database. And again, when this variant comes again in the future, maybe in another sample of your project, you will notify to see that, okay, so this variant has a review and I see it was important for my case because it has relevance on the clinical, etc. Last, I want to show you how the copy number variant results looks like. We usually get to see a circus with the representation of different SNPs of the patient and also a list of detected variants. By clicking on each of the variants, you can get to see a more detailed view. For example, on this picture, we are watching amplification. That's why, because we see like your sample is going up than the rest of the background. This is quite important because copy number variation detection could be really challenging, mainly on exome and panel data. And you really need to have a robust background so you can compare your sample with it, and that's not always easy to find. Now, taking apart all that things, I want to focus on what are the daily tasks of a bioinformatician. I think it's really fancy to show pipelines, to show results, graphs, and all the things we generate, but on the daily basis, we have to deal with some tasks that might be more repetitive, but they're really important. One of them is data comprehension. I think as a bioinformatician, it's really valuable to have a deep background on the biological field. That means you really need to understand the data. You really need to understand the genetics, the biological implications of the project, etc. Because sometimes an analysis don't go the way you expect, and you need to be able to analyze all the outputs you are getting and try to do it in a good way, etc. Documentation. I think traceability is really a key point, not only for others to try to follow what you have done, but also for yourself. It's really easy to come across a project 2 years later and don't be sure about what you did and why. So having the traceability of your projects and analysis is a key point. I suggest you, for example, to use Git for the code, but also inside projects have all the documentation and summaries, etc. Bugs detection and solution. I think there are always non-contemplated situations on your analysis. And sometimes, mainly when you have a big pipeline or you have a software, you will encounter situations where you really need to be a lot of time testing and trying to figure out what's going on, how to do it better, etc., to provide good solutions. You also need to stay updated with databases. Bioinformatics is really an evolving field. There are new things like nearly each month. So you really need to understand which databases you have, which ones you need, and inside the database, the information also changes. So you need to be able to extract just information that is valuable for your analysis. Also, and last but not least, data file manipulation and conversion. We've seen that in bioinformatics, there are well-known formats, but even though those formats have its own particularities, and for me, as a person working on industry, sometimes we have received files that don't really follow the specification they should. So you really need to be testing and trying to figure out how to work with it and to correct the format, etc. And now from a point of view of the industry, what are for me the key players? From my experience, I think in industry you really work on multidisciplinary teams. That means it's really difficult to be an expert on every field. I don't think that's possible. So it's good to be in a team where a person is focused on one area, another one is an expert on the other, and also to work into disciplinary teams. That means you, for example, in our case, that we are in contact with the geneticians of the company, also the IT team, and it's good to have meetings and project discussion because it's easier to find solutions together. The final goal of the company is always to improve the customer service. I think we have to keep that in mind. Maybe in academia, the final goal is different, but in this case, the products you produce or develop, they are going to be on that aim. Also, again, coming to the point I said before, everything needs to be reproducible and well documented. On a basis of an industry, this is even more important because you cannot come across a project from 3 years ago without being able to know what you did. You need to be able to do exactly the same if you are requested to, and also know everything that was done. There's a lot of important of validation and quality assessment. In our case, for example, as I said before, we have some quality certifications, but we also try to participate each year on external quality assessment like EMQN and GenQA schemes. Also, when you change something from your pipeline, some parameters, some algorithms, when you're trying to develop something new, you have to look for truth sets, you have to look for validated data sets to try to test and validate your pipelines. Also, you don't work on a unique project. With that, I want to explain that maybe you can do repetitive stuff, for example, the same analysis, but they will be in different projects. So you are always going to learn something new, and every project, they will have their own particularities. So you will have to maybe adjust some parameters or change some steps of your pipeline, etc. And lastly, to finish and to open a little bit the debate, I want to share with you this post of LinkedIn I came across the other day because I think it's really insightful. It says, it's nice to have the truly exceptional 1% of computational biologists who come up with groundbreaking tools and discoveries. But then we also need the remaining 99% of computational biologists to translate that into something useful for patients. I think that's really mimicking what is academia versus industry. And like Lee says, did someone come up with a really clever way to extract actionable insight from messy biological data? That's great. But who is going to study that paper, get their code running, do due diligence on the method, and tie the insights to actual decisions? That's what the 99% does. That's us. We don't all need to be the groundbreaking 1%, but we aspire to be the valuable realizing 99%. With that, I want to mean that it's not one thing is not better than the other. Both things are necessary, both things are valuable, and sometimes maybe in industry you don't develop a lot of new things, but you are going to study a lot and you're going to try to implement the best softwares and the best pipelines possible to help your patients or your clients in research. And that's sadly what Kristen Klinger is saying here. So I hope this was interesting for you. And if you have any doubts, you can put it on the YouTube channel and we will try to answer it later. Thank you very much.
Thank you, Alicia. Now we continue with Dr. Ali and we will open the discussion afterwards.
Thank you, Alicia.
Hi, do you have my voice and?
Yes, perfect.
Yes.
Okay, so I'm sharing my slides as well. You have my screen right now, yes?
Yes, of course.
Okay. So hello everybody. It's my great pleasure to talk in this community. So I want to talk a little bit about the aspects of artificial intelligence and machine learning in analysis of bioinformatics data. So whenever you have data similar to this one, that you have some benign samples and the other malignant samples, and you want to make a classifier in order to predict, for example, if some new patient arrives at the clinic, whether they are healthy or have cancer. So in some cases, the problem is very easy. You have a group of points from two different classes, and you can make a very simple linear classifier between them that is essentially a line or a hyperplane, and you put every new point on it and see whether if it is above the line or hyperplane, it belongs to class A, and it is below it, it would belong to class B. And you are looking for maximized margin of that classifier from each group of your points, and that's called a support vector machine algorithm that has been used for many years. But in many cases, the situation is much worse. For example, this is the real data from 10,000 different samples of 33 different classes of cancer plus one healthy group. And we wanted to know if we can find a classifier to distinguish between the points or the patients of each group of classes. So whenever we use the principal component analysis, like the left panel, it was very horrible. Whenever we use, I hear the echo of my voice, so maybe one mic is still, yeah.
I think I found unmuted, Professor.
Yeah,
Sorry.
thank you. Yeah, no problem. So whenever we look into other dimensional reduction algorithms like TSNE, we can see the points are better distinguished from different cancer types. So each point represents here a patient, and the color of each point represents the type of cancer. And still, with a dimensional reduction algorithm like TSNE, you see a mixture of different cancer types, and you cannot use a very simple classifier like support vector machine to distinguish between them. But whenever we take an idea from our brain system that can recognize much more complicated patterns, for example, nonlinear patterns, we can have better idea how to make models in order to classify more complicated patterns. So whenever you look at the synapses between the axons of one neuron and dendrites of the other neuron, you see there are some message passing between one neuron to the other neuron, and this message passing is done through synapses which are different in strength. So some synapses might work better, the other synapses might be weaker. And whenever you put some electrode array on the brain of mice or monkey or human, you'll see some sort of spikes that whenever they are more frequent, it means the neuron is kind of turned on. And whenever the spikes are less frequent in a unit of time, it means that the neuron is turned off, very similar to a transistor that is on or off. And you can take this idea as some sort of artificial neuron that has these synapses with the other neurons. For example, you see here the dendrites are the input connections into that neuron, and the weights represent the strength of those synapses. And you put a very simple linear function here. For example, W1 x1 plus W2 x2, etc., plus Wn xn plus some bias, which is just the a fixed number. Pardon? Is there any point? Okay.
No, just me.
Yeah. So you will have here some sort of very simple linear function, and but all of us know that this linearity means that if we add more and more neurons, everything is remains very simple linears, and it would not be able to distinguish between very complicated patterns. So we add a nonlinearity here, but by adding some nonlinear activation function. For example, you see this sigmoid function that if the sum of those values into some neuron is negative, the output of this sigmoid function is very close to 0. Whenever the total is very positive, the sigmoid function approaches 1, and in the middle there is some value between 0 and 1. And the good thing with this sigmoid function is that it is everywhere differentiable, and we can compute the loss function. And the loss means, for example, whenever you want to predict some input picture as a cat and the other picture as a dog, you assign cat to number 0 at the output and dog to the number 1 at the output. And whenever you enter an image of a cat and the output is 0, it's perfect. But whenever the output is higher than 0, this amount is considered as some sort of error. And we have these algorithms, error backpropagation, that you compute the effect of each weight on the output error and change a little bit each weight in a direction that the output error is slightly reduced. For example, you see increasing W0 by a little bit would increase the error. So in that case, you might decrease W0 by a little bit. And this is in more details done by computing the partial derivatives of the error with respect to each of W0, W1 until Wm. And whenever we want to compute these partial derivatives, we use this chain rule. I don't go into the details of math here, but this algorithm is very effective in increasing the speed of finding optimal values of W0 to Wm, which are called the parameters of this network, in order to minimize the errors on some input data. And this is called finding or learning the values of these parameters based on some data is called machine learning. We are learning the weights. So we can put these neural networks in a very deeper way. For example, rather than having only one layer, we could have 10 or 100 different layers. And this is very similar to how our brain works. And a very specific type of neural network is called autoencoders. And these autoencoders are very useful in bioinformatics in the sense that they do not need any labels on the data. So we can crawl and download millions of different samples from gene expression omnibus or similar databases, and each gene expression profile is a vector of, for example, 20,000 different genes, and we can input this vector as the input of the neural network, and we expect the autoencoder to reproduce the very same vector at the output. And this problem is obvious whenever we have enough neurons in the middle, but whenever we have a bottleneck layer at the middle with very few neurons, it's very hard to compress and take the very main part of the information of gene expression profile at very few number of neurons at the center, and to decompress or decode the whole data out of it. And one application is, for example, you have this autoencoder, you provide some picture, you compress it in a very few number of neurons at the latent vector in the middle, and you want to decode the whole image out of it. This is one kind of this autoencoder. And we used this structure of autoencoder with 20,000 neurons, each neuron representing the expression value of one gene in the beginning, and we reduced the number of neurons to 2,000 in the middle, and then to 30 neurons at the bottleneck layer, and we decompress the whole thing at the end. So by giving this network the raw expression values of like 60 or 64 different tissues of our body, nothing more, nothing, no data about the genes and what is the name of these genes and what are the pathways or gene ontologies. We just provided the expression profiles at the input, and we expected the network to reproduce the expression profiles at the output. But we eventually identified that each of these neurons, we had these 30 neurons in the center, and each of them was learning a different aspect of cellular biology. For example, whenever we triggered one of each of these 30 neurons in the middle by increasing its weight or its value a little bit, we notified different sets of genes at the end were triggered. And whenever you use these gene ontology or pathway databases in order to annotate those set of genes, you would understood, for example, the neuron 3 will trigger some intracellular vesicles, while neuron 11, for example, was linked to a spindle and cellular division, and another neuron to for example, cell surface, another neuron to like immune system, etc. And that was very interesting for us. We submitted this paper on September 2017, and it was accepted more than 1 year later in 2019. And it shows that in that time, how careful was everybody in the biology about these deep neural networks. So people did not believe the outputs of deep neural networks very well. But at the same time, we started another study to use an extended version of these autoencoders in order to solve multiple instances of tasks. For example, in addition to encoding and decoding the mRNA expression profile, we wanted also to classify or to recognize the tissue of the cell or sample out of the bottleneck layer. So we connected this bottleneck layer to additional neurons, and we try to predict the class or the tissue of the sample out of it. And in more detail, so it was a multitask learning architecture. We provided mRNA expression profile as the input of the network, and we expected the tissue type, the disease type, the mRNA expression profile, and the microRNA expression profile to appear at the output. And we observed that there is a very great tissue classification. So everybody might know about these type of matrices, which are called the confusion matrix. So in each row, you might see the number of samples that are actually belonging to each class, for example, to each tissue, and the columns represent the predicted classes. So, for example, for the ovary, if you see the whole row, there are 376 samples in total for the ovary, which all of them are predicted as ovary. There are very few non-zero items, for example, 3 cervix samples that are um, mislabeled or mispredicted as head and neck. And this is the classification accuracies or confusion matrices for the disease. So each brow or and column represents some class of like cancer. And you remember this picture from the beginning, but whenever we used those very few neurons of the latent layer or the code layer in the middle, rather than the whole gene expression profile, you observed and you use TSNE on top of that. You observe the right panel here that the samples from different classes of cancer were very distinguishable. So the left panel is using the PCA on the same data. The right panel is using TSNE. And it was also submitted and published in scientific reports of Nature in 2019. And if you are interested, you can refer to this article which we named Deep Pathology. And I want to introduce a very recent work. How much time do I have?
We had 20 minutes, so it should be finished right now, or 1 minute maybe.
Okay, so we also use the same architecture of deep neural networks in order to predict what kind of non-coding, small non-coding RNAs are secreted from a cell in the form of exosomes, and what class of them are not secreted. So whenever you look, so these exosomes are a very small package of like a membrane, which is very similar to cellular membrane, and they contain some very tiny molecules like parts of nucleic acid, very tiny RNAs, very short fragments of DNA, and also some proteins, some and membrane proteins, and but they are very important. For example, in cancer biology, they can carry messages from one cancerous cell to the whole cancer niche and increase the like angiogenesis or epithelial to mesenchymal transition. So our question was, whenever you look at the cancer cells, you will observe there are non-coding RNAs that are secreted, and the other class of non-coding RNAs that are not secreted. And the question was, what is the mechanism behind selection of which non-coding RNAs are secreted or not secreted from the exosomes? So we used very different, simpler machine learning algorithms, but eventually we started using these gated recurrent units, and these were just before transformer attention mechanism that are currently used for large language models. But whenever you use these gated recurrent units, we achieve the highest accuracy is in comparison to simpler machine learning algorithms. And we were also able to predict which mutations make a big change or big impact. For example, we had these non-coding RNAs that were not secreted from the cell, but as exosomes, but with a very specific mutation, one single mutation, our algorithm predicted that they will be secreted. And whenever we use these predictions and went into the lab and designed the same mutation, we observed that, okay, our prediction was correct. So the mutations, the single mutations on the non-coding RNAs that were not previously secreted in the form of exosomes, with that single mutations, they started to be packed into exosomes and secreted from the cells. And whenever we observed, okay, what is the actual pattern that our gated recurrent unit is learning in order to classify which non-coding RNAs are secreted or not, we noticed there is a very similar pattern with some of the RNA binding proteins. And we designed some novel experiment called ExoClip. And whenever we tested them using this technique, we observed, okay, that's true, that these non-coding, these RNA binding proteins actually play a role in this secretion. So it was published in Cell Genomics recently, and it was in collaboration with Dr. Hani Gudarzi of UCSF University. So I wanted to thank all of my labs. I do not have the pictures of all of them here, but this is the most recent picture of the people of my lab. And thank all people for your attention.
Thank you, Professor Sharifi, for very detailed insight and very interesting examples of bioinformatics in academia, and we were so interested to know about them. And then we can start the question and answer. And please, everybody, if you have any kind of questions, any kind of doubts, please ask Professor Sharifi and dear Alicia.
I want to do an icebreaker question to Dr. Ali. I was wondering, given your trajectory and the difference of years that you have been in academia, and especially now that you are working with machine learning and artificial intelligence methods, that I was wondering two things. First, do you think that you would have used companies? Imagine that a company is offering you one of their services for free, one analysis for free. Would you have used those analyses from a company, or would you have preferred to use one service or like a nature method instead, a common pipeline instead of a private company analysis? What would have you done back in your day?
Yeah, if I am a bioinformatician, I would analyze the data myself. If I am not a bioinformatician, I would learn bioinformatics, because nobody is more careful about my data rather than me, and nobody is more enthusiastic in analyzing my data than myself. So this is my idea. This is my idea that the biology, genetics, and medicine people who are interested in like cellular and molecular biology and want to conduct research there they really need to learn bioinformatics, because in the very same way that they need to learn how to use microscope. But the era of observing or deciphering new aspects of cellular biology using microscope is mostly past. And nowadays, the actual microscope is bioinformatics. So I have seen better company services, and I have seen terrible company services. For example, there was some of my colleagues who sent the data to a company for a bioinformatics service, and I noticed a very initial and obvious mistake at the very beginning of the analysis, and the whole rest of the analysis was based on the very beginning. So the whole thing was kind of very mistaken. And I would say I would trust the data that analyze myself.
Okay. We have one question from
Thank
the
you for your
audience.
answer.
Maybe we will go with that first, and then I will send you the second question, Alicia. Edward Blake.
Hi. Yes. Can you hear me?
Yes.
Perfect. Okay. I'm doing my master's in bioinformatics, and next year I'm going to be starting to look into the industry. But since it's such a massive field, which sector should I go into? Like, I know, of course, AI is like the big keyword, but it seems like today it's just AI is just another add-on to what other fields you have to go into. Would you know any fields that would be recommended?
You can give maybe an insight, Alicia. Yep, yeah.
So I think it really depends. Like, we have seen bioinformatics really huge. For example, I think is from the genomics point of view, I think it's gonna get a lot of insights, long reads, how to analyze long reads, for example, I think is going to be more important in the future. And maybe from other perspectives, also there's a need to metagenomics, because we are suddenly getting so many samples. And also RNA-seq, like, I think is really, really important, mainly on single cell and spatial genomics. As you can see, there are several fields, nothing to do one into another, but I think you just need to start with one and try to get like the basics, because one, you get like the knowledge of one of those fields, it would be easier to start learning others, and you will see how the industry is moving and adapt yourself to that. That's my personal opinion.
All right. Thank you.
I would recommend you, Edward, to follow RSG-Iran or any other RSG, and you will get or grasp the feel of what's there in the industry. That's why we are in these communities.
Perfect.
And
Will do.
Anybody else on questions, or shall I drop the second one? Do you have one, Hossein, maybe?
Of course, yes, because I can see a lot of students among us in this session. And I want to ask Alicia, which kind of certifications or skills students can learn and obtain before they want to apply for jobs in the industry?
Well, I would say from a student point of view, that a good starting point would be when you need to do like your dissertation, like bachelor dissertation, master dissertation. It can be really useful to look for companies. Like that could be like a good starting point. But if not, imagine you just do it at university or academia. I think it's useful to try to contact a lot of people. You are going to get a lot of no's in the process, and a lot of people won't even answer you back. And some of them will tell you, okay, it's fine, but we don't need you right now because we want more experienced people, because I also got that. But in the end, new opportunities arise. You will see some trainee roles, go for that. Also, I don't know how it's in Iran, but in Spain, we have scholarships that, for example, you can be a researcher for 2 years, like a technical researcher in different groups. And I think it's a good point also because you get to see what is academia like, what is research, and then you can make your decision if you want to take that skills you have learned and try to apply it for a job at the industry, or just keep going and turn into PhD maybe in the academia. About the principal skills, I think you always need to know Bash. That's you will need for everything, absolutely. And also about the language, you can start with R or Python. It depends, because you can get into a company and maybe they will just work on Java. I don't know. So I think the most important is to have the basics, and like they see you're really motivated and you're open to learn new things and try new things. So I would say that would be.
Great, thank you. And just one very short question. Which platforms do you normally you're using for contacting people, LinkedIn or by email or any other kind, I don't know, to find people in different companies and contact
Yeah,
them?
I think LinkedIn is really useful, not just to contact people, but also to, as Adrian said, you need to follow groups of bioinformaticians or industry. It's useful to go to this type of events, also presentially. And also there are some clusters in each country, I guess, at least here in Spain, that they group several companies from the biotech industry, from the medical industry, and there are sometimes you have some emails so you can get to contact them. It's not so easy to find like all the companies that are there, and maybe also trying to find spin-offs coming out from the university could be useful.
Do you
Thank you.
agree, Dr. Ali, with what she has said in regarding the profiles of young bioinformaticians?
Yeah, I would mostly use Twitter in order to find what's happening. For example, and it really depends on the field of a study. So many people in different scientific communities might use different types of reaching to each other, but yeah, it's I would say it's always also depending on your target, but it's good to have a look at the latest conferences. For example, in the bioinformatics, we have RECOMB and ISMB conferences, which are great to look at the latest articles and to know what is the trend, what is the hot topic, and also to be there to actually meet the other people. So it's much better, I think, rather than just sticking into the social networks. But yeah, social network is also good.
So I'm going to continue, if you allow me, Hossein, because I had one question, and actually someone from the audience in YouTube also wonders the same as me, and this question is addressed for you, Dr. Ali, and is that we see that many of the current bioinformatics analyses are based either in machine learning or in artificial intelligence. So when you perform such analysis, I was wondering, and how do you validate them? I mean, do you use, for example, you haven't talked anything about explainable AI, in artificial intelligence. I mean, how do you validate your methods based on machine learning and artificial intelligence?
Yeah, that's a really good question. So whenever you observed, for example, the very initial slides of my talk about those autoencoders, whenever we identified that triggering each of those neurons in the bottleneck layer would trigger a different set of genes, and those genes were really related to each other in terms of cellular biology. For example, many of them were working together in a pathway or in a gene ontology or in a very specific compartment of the cell. So this is kind of explainability. So this shows that we are not in the wrong direction. So our neural network is really learning some aspects of cellular biology. And for each problem, there is another way of explainability. But in order to have a better understanding or confidence about how your network is running, there are general techniques. For example, one technique is to separate your training, your validation, and your test data. And this is very important, not to have a leakage from training to test. And this is very often that we observe in, for example, papers of bioinformatics using artificial intelligence, that there is an indirect leakage. For example, you see a paper that is going to predict the binding affinity between some, for example, MHC, the major histocompatibility complex, and peptide. And you see, okay, the paper has splitted the data between pairs of MHC and peptide in the training and different pairs in the test. And you might guess that, okay, this splitting between train and test data is already done. But whenever you go deeper into the data, you will see the same peptide is in connection with some MHC, is in the training, and in connection with another MHC is in the test. And the fact is that some peptides are less willing to bind to any kind of MHC, and some peptides are more willing to bind to any kind of MHC protein. So whenever you have this peptide both in input and in the output, you are leaking data from your training set into your test and validation set. And the correct way of doing it is to completely split the data in order to ensure that there is no peptide in both train and validation or train and test sets, and there is no MHC protein both in train and test set. So this is kind of it. Or I can bring you many different common mistakes. For example, whenever you have imbalanced data, let's say 99% of your samples are from healthy people and 1% samples from cancer. And you will achieve accuracy of 99%. But that might be the case that all of samples, but by your machine learning algorithm, are simply classified as healthy, and your algorithm is simply useless, but it is producing 99% accuracy just because of this imbalance in your data. So I wanted to bring you many different cases that ensuring the validity of the or explainability of your machine learning algorithm is not the same in different problems, and mostly is achieved by some sort of studying similar papers or similar researches plus experience.
Nice. And I was wondering, given this answer, Alicia, that do you think that what are the chances in a near future that companies start adopting these methods to sell a product based in these methods? Because At the moment, the only companies that I know that use such methodologies are those as the one of OpenIA with AlphaFold, and only ones that actually have a large volume of data. And also thinking on the difficulties of, for example, in health, that you required a lot of legal licenses. How can an artificial intelligence be framed in a current company?
I'm not sure about how it's going to evolve, as you said, but as I explained, like in my talk, I think the problem we have is that we need that kind of validation. We need to be like sure. I saw no, right? And we need to be sure about what we are using. And with that, I mean, maybe it's not the case, but for example, it's kind of easy to find new tools, new algorithms, and new things. And when we try to implement it, maybe they were not actualized in 4 years ago, 5 years ago, and we kind of need to use updated algorithms, mechanisms. We need something that we really, I don't know, like there are a lot of tools and there are plenty of possibilities. So I think that's the main problem. But I agree with you that there's a need on the industry and maybe we will evolve to that. Also, as I ended my talk, I think research has to do a lot there. Like maybe we cannot focus so much on that. Maybe some companies do. For example, as you said, I think it's Google who developed a new variant detection tool that is focused on artificial intelligence, I think. So there are some companies that are really focusing on that, but maybe for the smaller ones is, I don't know if easier or more convenience, like to work on more validated things first and then try to apply those things.
Great, thank you. Also, we have a question in chat box. The first part, it seems related to academia, and the second one is related to industry. Hani asked, what strategies do you recommend for generating innovative ideas that can be effectively implemented in industry, which is the first part. And the second part is, once we have these ideas, what steps should we take to turn them into practical, impactful solutions, which could be the industrial part?
Okay, so I can, from my limited experience and what's in my opinion, I think first to understand what their needs on the population. If you are focusing, for example, on direct solutions for patients, like, I don't know, in diagnosis, so maybe you really need to understand what's a concrete need. I don't know, for example, on genomics, you can see that there's a gap on detecting copy number variants on genomes. So maybe you need to try to have a research to see which types of algorithms that are available, which are the gaps, which are the strengths, which are the weaknesses, and you try to develop something that overcomes those. That focus on the area I control a little bit more. But I guess it's the same with RNA-seq or machine learning. Like you really need to understand first the problem because it's easy just to say, like, try to focus on something, but maybe later it's not so easy to use it or to have like an application of it. And of course, you have developed something that is great, but maybe no one is going to use it. So for my opinion, that would be like the main point. About the second option, I'm not sure how that would go. I guess kind of the same, like if people really see an interest on that, like you will have the publications and maybe like people have a look at what you have developed, they will try to replicate you, they will mention you, and then that new tool will start being used. Maybe Dr. Ali could add something on that part because I think it involves both academia, well, mainly academia because that's where it's gonna be developed mainly.
Exactly.
Yeah, only thing that I want. So your answer was great. Only thing that I wanted to add to this meeting was thanking my prior supervisor from Spain. So I met him during the time that I was in Max Planck Institute in Münster. So he was Professor Marcos Arauzo Bravo, who is currently a professor as Biodonostia, if I have spelled correctly, in San Sebastian of Spain. So I wanted just to thank him. Yeah.
Thank you so much, Dr. Ali.
Thank you.
So do we have time for more questions? Maybe one question that maybe also goes in concordance with your last answer, Alicia. Dr. Ali, do you think that there has been a growth of bioinformatician companies over the years?
So I would say the aspects of bioinformatics are increasing. For example, in protein design, in many previously locked aspects, because recently we had a major innovation, which is Google DeepMind, Google like AlphaFold, DeepMind AlphaFold, which won Nobel Prize. So a problem, a very central and very important problem that was locked for many years, was solved in 2021. And out of it, I would expect that many different companies would have different innovations in expanding our therapeutic alternatives, etc. But I would say those companies might be also a mixture, not only from bioinformatics, but also in bringing that products into real products rather than just an analysis company. Yeah.
Thank you so much. Anything more to add, Hossein?
No, thank you. From my side, no. I don't know if other audience have any question regarding these topics.
I mean, I could be talking to you a lot because it's very interesting, the different profiles, and I think we can learn a lot from you. Indeed.
Yes, I can see a lot of people with different profiles, but since nobody is asking any kind of questions.
So I think that then we can conclude, right, Hossein?
Yes, of course.
Thank you so much, Dr. Ali, for being here with us, for making possible this collaboration between the two RSGs. Also to you, Alicia, for this challenge for you. I mean, being in front of someone like Dr. Ali can be maybe overwhelming. So thank you so much for accepting the challenge. And thank you, Iran, RSG-Iran, and every one of you for accepting this type of activity. Thank you also to the Spanish Society of Bioinformatics and Computational Biology, which give us the medium to do this type of activities. And of course, to the audience, and I encourage you all to be part of the RSG's communities, because you can never know when you are going to learn something useful for your life. So,
and shortly, I want to say thanks to Professor Sharifi and also to dear Alicia. And we used it was very useful to share their experience with us, and also the RSG-Spain for all their efforts in this collaboration. It was an honor for us and from all the audience here. Thank you very much.
I wanted also to thank all of the organizers from two RSGs, and a special thanks to Alicia with very nice presentation, Adrian and Hossein for very nice hosting this event, and any other people who was involved in organizing this meeting. Thank you very much.
We also want to say thank you, and also to you, Dr. Ali, because I think I really learned a lot from your presentation as well. So it was really inspirational for me. So thank you very much for all.
Bye.
Thanks, sir. Goodbye.
Everybody, thank you.
Bye. Bye.
Bye. Thank you.